So I want to pick up with a bit of an overview before we move into talking about the individual groups of plants, kind of giving you an overview like I often give you a phylogenetic tree. Only instead of a phylogenetic tree, this is sort of a grouping of plants. Last lecture, I said that the term phyte, P-H-Y-T-E, refers to plants, things that are related to plants. And so at the top here, we have the big category that includes all of the green plants, the streptophytes. And if you remember when we were looking at our phylogenies for the protist groups back when we were sort of in our earlier organisms, plants were just in there amongst the other algae on this one branch in the tree of life. And so plants are really closely related to a lot of the algae. We have some ideas about which are their closest relatives, but they share a lot of common characteristics with some of our single-celled eukaryotes or our aquatic eukaryotes in the algae. Probably their closest living relative, as of now, this is changing a little bit, are the charophytes, and those are aquatic plants. And then over here we have the embryophytes, which are the land plants. And we break the land plants sort of into those plants that are vascular and non-vascular, so whether or not they have a system for transporting fluids throughout their bodies. And then we also have those plants that are seedless. Some of them are non-vascular and some of them are vascular. And then the seed plants. When we think about this from a phylogenetic perspective, like gaining vascular tissue is a more recently evolved trait because it's associated with adaptation to drier and drier land environments. So moving into more arid and desert land environments. Seed plants are also an adaptation for being able to live in a more arid or drier land environment. Okay, so with that context, I'm going to talk just really briefly about green algae, and then we'll move on to talk about the major groups of plants. So green algae are all in the group known as charophytes, and they are all photosynthetic eukaryotes. So eukaryotes who carry out photosynthesis, they're autotrophs. They're going to be using sunlight to produce their food, but they have complex multicellular bodies. So we sort of differentiate them a little bit from our simplest of green algae, most of them having multicellular bodies. They store energy as starch, very much like our plants do. They have cellulose in their cell walls, and they use the same pigments that land plants use to carry out photosynthesis. So really, they're the aquatic analogy to land plants, right? They have a lot of those same features. So we looked at some of these organisms when we had our protist lab. We saw some colonial spirogyra, like this guy here. We may have seen some chlamydomonas that were mixed in with other groups. Here's ulva, which is a common algae that you see on coastlines. You might see this if you were to be out on the Cape, on the shore. If you're somewhere where there are tide pools and rocky shores, you might see members of this species. The species that has long been thought, or the group that has long been thought to be the closest living relative of land plants, are the charas, the trals. And so this is a group of charas. You can see that they're under the water. They are living in the substrate of shallow water. This box up here kind of shows you zoomed in on a single individual. They're different from land plants in one key way. They don't have alternation of generations. So you don't see this alternating haploid and diploid life cycle we talked about on Tuesday. And then they also differ from land plants in that they don't have a lot of the physical features we think of as being key to land plants. Like they don't have roots, they don't have stems, their leaves are not true leaves, and they don't have analogous reproductive structures. So they're more analogous to algae in that way, in their body plan. But they seem to be the closest relative to modern land plants in terms of their other features. Although recent genetic analysis has determined that maybe Spirogyra is actually closer genetically to land plants, even though these guys are closer maybe in some of their characteristics in terms of what kind of structures they have in their body and their physiology. So assuming that we have some sort of ancestral green algae that's maybe like a chara, maybe the line that gave rise to something like Chara, we move from an aquatic lifestyle to all of these groups over here on the right, which are land plants. And so we have some of them are very small. You might not even notice them if you didn't have a hand lens out or you weren't crawling around down on the forest floor. Things like liverworts and hornworts and some of the mosses are all really quite small. And then we have club mosses, get a little bit larger and more evident than ferns and horsetails, and then gymnosperms and angiosperms. And so kind of along this stair step, all of these groups, liverworts, hornworts, and mosses are all non-vascular. And then liverworts, hornworts, mosses, um, ferns, or club mosses, ferns, and horsetails are all seedless. And so it's not until we get down here where we start to see sort of true vascular tissue in the ferns and then seeds and pollen in the gymnosperms and angiosperms. We have sort of really different characteristics of those plant bodies. The club mosses have some vascular tissue, and they might, I get, it looks like this branch here. I don't, I don't think they have lignin. I don't think they have true vascular tissue, but they have some transport. Um, I'd have to, I have to bust out a diagram and look at their anatomy again. I don't think that they have true vascular tissue. Okay, so the first group that I want to talk about a little bit are the bryophytes, and the bryophytes are the seedless non-vascular plants. And they tend to require moist environments, particularly in order to actively grow and thrive and to reproduce. That being said, most of them also have some ability to be desiccation resistant or to be able to survive drying out and can sort of reanimate when moisture comes back. And so in this picture, maybe if you can use this moss as a reference, all three of these, this is a highly sort of zoomed in image, right? If you were to walk past a liverwort or a hornwort in the forest, you would just see some nondescript green patch, and you would have to use a hand lens or zoom in a little bit in order to actually see the structural shape of these organisms. The liverwort gets its name because it has these big leaf-like structures that actually aren't leaves that look kind of like the lobes of the liver. They have this sort of rounded shape, and so that's where their name came from. Then we have the mosses down here. You guys looked at some mosses a little bit closer at the Botanic Gardens. In sphagnum bog, the sphagnum is actually a type of moss that creates that floating mat, if you guys have ever visited a bog. I don't know how common they are. I know both of them exist in the eastern deciduous forest. Um, I have not paid enough attention, and I don't know that I've ever encountered one in the wild. Yeah, yeah. I think in our minds we probably, yeah, if you have a hand lens on you or you've got like a loop, zoom in and look. I'm pretty sure I've seen a liverwort in the wild. But I've probably spent too much time walking past them not looking carefully. Okay. I have put in this table from another book because I think it just gives you some useful summary for these different groups. So the non-vascular plants include three main groups of organisms: liverworts, hornworts, and mosses. In all three of these, the gametophyte is dominant. So when you see one, when you look at it, what you are looking at is the gametophyte structure. Okay. You might see a sporophyte if you see like the spore forming structures of the moss. And actually, hornworts will get these bright red structures when they're producing spores. And so you can see the sporophyte on them a little bit more easily. Um, and that sporophyte is going to develop from a zygote. So you're going to have fertilization to produce a zygote, and then the zygote will grow the sporophyte sort of from the surface of the gametophyte itself. Okay. Um, they all have motile sperm, so they have flagellated swimming sperm. They have to be able to travel to the egg. So that means that moisture is required for reproduction, whether that's a heavy coating of morning dew or an actual rainstorm. They require some surface film of water in order for reproduction to occur. The egg is retained on the gametophyte and is stationary, and the sperm travels to the egg. Early embryonic development occurs within an archegonium. We're going to see one of those in a minute. And they move via haploid spores. They're carried on the wind. The spores then will germinate and grow and produce the next gametophyte. They don't have any structures for transporting water and nutrients, and they actually don't have any roots, and they're going to get their moisture just from absorbing it through the surface of their tissue. So the whole hornwort or whole liverwort or moss can actually absorb moisture and bring it into the body, moving it through diffusion. Okay, so some of these things I just said in that table, they lack true roots and leaves. They have structures that are maybe leaf-like in appearance, but they are not at all anatomically the same as the leaves that we see on higher plants. Um, they don't have any lignin, which is a structural support chemical in their cell walls. And so that's one reason that they're small in size. They're also small in size because they don't have a way to transport water. And so they have to be kind of low to the ground where they can harvest water. They don't have the ability to move water from the ground up into their body, and they don't have roots to bring water in from the soil into their body. And so they're limited to sort of that contact layer low on the ground or on the surface of a tree or a rock or something like that. Um, they're desiccation tolerant, and many of them, especially the mosses, are known for their resurrection abilities. And so they actually have a hormonal trigger when the land starts to dry up and moisture starts to go away, and they will basically put their bodies into a like hunker down state that is similar to how the embryo in a seed actually prepares to be left from the parent plant and kind of stagnant in that seed until germination occurs. And so they pull in their resources, they protect their bodies, and they can actually become like crumbly. Like you can have moss that's dry that you can crumble it in your hands. And then if you reanimate, you add water to it, it'll reanimate and fire up photosynthesis immediately. And so like as it starts to rebuild its structure, it fires up photosynthesis. That process causes a lot of cellular damage to the body, but the photosynthesis provides new resources, and as soon as it has new resources, it basically rebuilds itself and sort of can resurrect. So as long as they have, sufficient resources left in their quiescent state, they can reanimate when water comes. And so some of these actually live in pretty dry environments, like on the tundra, where we don't have a lot of liquid water. All the water is frozen, and they're able to get through those periods with no liquid water by kind of hunkering down, and then they reanimate when moisture is available, when things thaw. Okay, so bryophytes have a structure known as the gametangia, which is going to be what produces the gametes. And so this is going to be the archegonia and the antheridia. The first word here, archegonia with the IA and antheridia with an IA, is plural, and the is singular. So these two terms are not necessarily different terms, it's just a singular and plural for the same thing. Okay. The archegonia are reproductive structures that in which the eggs develop. And so if you were able to actually zoom in and look at the tip of certain species of moss, you could actually tell whether you were looking at an egg producing moss or sperm producing moss based on the shape of the tip of the moss. And so the egg producing moss, the archegonia, is going to produce an egg in the cup of the tip of the moss. And the antheridia, the male producer or the sperm producing moss, are going to produce sperm in that same place. When the sperm are produced and there's moisture available, then the sperm will swim off in the beads of moisture, in the dew, in the rain, and head towards the egg. And when the egg is fertilized, it's going to actually produce the sporophyte. And so in this gametangia system, we have separate haploid, sort of male or female individuals, egg producing or female or sperm producing individuals. The egg remains with the individual, is not free moving. The sperm swims over to the egg. This means that they have to have moisture for reproduction to occur. So if there's a long period of drought and desiccation and no moisture, then no reproduction happens. There's no growth, there's no reproduction, they're waiting for moisture to come back. The fertilized egg is going to produce haploid spores via meiosis. And so the sporophyte structure is going to be cranking out haploid spores. Those spores grow. I'm going to actually use this slide. Those spores are going to be produced in this sporophyte, and they're going to develop in this little capsule. And when the spores are completely matured and they're ready to be released, the capsule actually dries out. And as it dries out, this little end of the capsule pops open like a flip top on a cap. It's called the operculum, and it just flips open due to the drying out of the capsule. And then the spores are air and windborne. They can take off and move to a new location where the spores will then become a new gametophyte. So the spores are going to grow up entirely by mitosis. There is no fertilization required. Every single spore can become a new gametophyte. And then we start the cycle over. Okay. So just like we talked about alternation of generations in the last lecture, in the bryophyte lifestyle, as exemplified by this moss, about half of the life cycle is spent haploid, and the other half of the life cycle is spent diploid. Ability. Yeah. When we think of the resurrection, we're thinking of the gametophytes, actually. The spores, when the sporophyte dries out, it does that whether or not moisture is present. Like, even if moisture is present and the moss is actively growing, eventually, after the time period required to fully mature the sporophyte will dry out and pop open. And so that'll release the spores, regardless of whether they're in a desiccation state or not. Once that occurs, if the gametophytes are really dry, then there's not going to be the next sporophyte. Like, we're not going to have another sperm swimming to an egg to produce the next sporophyte. And so once the sporophyte is released its spores, the only way we get the next sporophyte is to have active reproduction, which requires moisture. And right. So that it can dry out in the air. And also, you want to be, if you want your spores to be dispersed on the wind and get a little further than the dewdrops are going to carry you, then you have to be somewhere where you can get a little bit of air movement to pick them up and move them. Yep. Okay. Then we'll move to the next group, which are the seedless vascular plants. And so these are called tracheophytes. So that term tracheo, like your trachea, is a tube. And so the tracheophytes have a tube-like system through their body to move water and nutrients. And they have these water conducting structures. So we call them vascular plants. They're vascularized. It's like they have an artery and vein system, except that it's xylem and phloem. So the vascular plants include club mosses, horsetails, and ferns. That's the first group we're going to talk about, and then also the gymnosperms and the angiosperms. In all three cases, the sporophyte is the dominant structure. So when you look at a fern, you're looking at a sporophyte. When you look at a wildflower, you're looking at a sporophyte. When you look at a tree, you're looking at a sporophyte. And in these groups, the gametophyte is microscopic. It's a tiny little cluster of cells, a small section of tissue embedded within the inside the sporophyte's body, and you would never see it without dissection in most of these cases. The fern is an example. You can actually see the gametophyte in this subgroup, but it's not something you normally encounter. Again, you'd have to be down crawling around on the forest floor with your hand lens to stumble across them. These guys are really completely microscopic. You're never going to see them. There's kind of a line here in the vascular plants. Club mosses, horsetails, and ferns still have motile sperm, so they have to have moisture so the sperm can swim to an egg, whereas the gymnosperms and the angiosperms no longer require a swimming sperm. They instead produce pollen grains, which are carried on the body of pollinators or on the wind. Embryonic development occurs in the club mosses, horsetails, and ferns in a gametophyte, and the gymnosperms and angiosperms in a seed. And here we have haploid spores for dispersal versus seeds and fruit down here. So there's a pretty big line here, but I put the whole table in with this one group so you can use it for comparison. Okay, so I like this picture of a leaf showing just the vascular structure in a leaf. And so the vascular tissue is just the tubular transport system that's found throughout the plant's body. So if you think of just like having a leaf in your hand, maybe you're imagining you're holding an oak leaf and there's a strong vein down the center of the leaf. We make it a maple leaf like my hand, right? You've got a strong vein down the center. You can actually see the ridge. You could almost fold the leaf in half, right? That's this main structural support for vascular movement. We've got a lot of fluid coming from the rest of the body of the plant out into the leaves. And just like your arteries and veins, the vascular system branches off of this large tube in the middle into these smaller veins. And so those are the veins that you would see going out through the leaf, and then into even smaller structures where we are diffusing out to the tissues. So now you no longer have to rely simply on osmosis and diffusion to move materials through the body, but you have a plumbing system of the body to move materials through sort of larger opening tubes. Those tubes and plants come in two different types, the xylem and the phloem. And so this is a cross section. So like I cut through one of those veins that I was just talking about. I cut my fingers off of my maple leaf and looking in, right? And so what you see here is what that sort of main large vein-like structure. And all of these cells that are outlined in red are xylem. They're large tubular cells. They're like straws or capillaries in the plant's body, and they move water and nutrients. And so they're large in size because they have bulk flow. They're non-living tissue. They're no longer alive. And the cells themselves actually fuse together with some plates in between, so you can create these long tubes. And if you are looking at something like a tree, those tubes running all the way up the entirety of the trunk of the tree, connecting, moving water from the roots all the way up into the top. And then the phloem is sort of surrounding the xylem in green here. And those phloem cells are also non-living, and they transport sugar, proteins, and solutions. So when you tap into a sugar or a birch tree to harvest sap, you're tapping into the phloem where the sugar solution is moving, and that's what you're trying to harvest, because that's all the nutrients and the stored sugars of the plant's body. Okay. So this system allows the plant to move materials over a much larger distance. So all of a sudden, the plant is no longer obligated to be small and low to the ground. It can get taller because it has a way to move materials other than like slow and ineffective osmosis and diffusion. Right? So the nutrients in the xylem tend to be small inorganic molecules that dissolve, like small ions, and little things like that, magnesium and phosphorus, things that it needs that are essential. But the bulk of the xylem fluid is water. And there's just some small dissolved substances in it, some solutes that get transported in that water. Whereas the phloem, the bulk of the phloem solution is also water, but it's very highly saturated with sugar. And so you have this is this really nutrient-rich food source in the phloem tissue. And then we also see, as we move into these plants with vascularization, we see lignin. And so all plant cells, even our single-celled algae cells, have cellulose in in their cell walls. But lignin is an additional chemical that is kind of like girders between the cellulose to reinforce the structural components of the cell wall. So this square here is showing you an image of a plant cell wall. The plasma membrane is here. So the plant cell is all over here in this space. It's not shown. Here's the plasma membrane. And then out here is the outer sort of boundary of the, the cell where we would have the epidermis type edge of something. And in between the plasma membrane and the outer boundary of the cell, we have these different layers of the cell wall. And in these different layers, all of this green is cellulose. So the cellulose is providing rigidity and structure to the cell wall. And then all of these little grayish, whitish shapes are lignin. And so the lignin is like scaffolding between the cellulose, and it reinforces that structure and makes it much stronger. And so lignin helps the plant to stand more upright, to have stronger cell walls, to have taller structure, and to ultimately reach a larger body size than it could if it didn't have lignin. Okay, so then we move into the vascular plants. Because of these things, we now have plants who can have larger body sizes and who can live in drier environments because they're able to transport fluids through their bodies. So all of these things represent vascular plants. Here we have some lycophytes or club mosses, some ferns, a gymnosperm, and an angiosperm. So a cone-bearing plant and a flowering plant. So they're able to live in drier environments. And the first group that we'll talk about finishing out this chapter are the seedless vascular plants, the tracheophytes, where we started here. And the tracheophytes include two main groups of organisms, the lycophytes, which are club mosses, and the pterophytes, which are ferns and their relatives. So in the center here is a club moss, and this club moss is producing spores in this strobulus, this beige colored structure here. We've seen these multiple times already in this class. I pointed them out to several of you when we were out at Masson Ridge, and they are also in the GCC forest. You'll run into them. Frequently, people think that they're baby pine trees. Baby pine trees can look similar to club mosses, but that's their full size. So they're maybe only just a few inches tall, and you'll see them in the forest floor. And so these are all the club mosses. And then over here we have a fern. We have lots of species of fern in this area. GCC's forest will see mostly Christmas ferns and woodland ferns. If you are currently foraging for fiddleheads, you're looking for ostrich ferns. There's lots of ferns here. And when you are looking for fiddlehead, these are fiddleheads. I just love this picture because it looks like this grasshopper's riding a bicycle. It's actually just sitting on fiddleheads. The fiddlehead is the young frond of the fern as it develops. So it's ultimately going to unfurl and just become, this leaf made of many, many leaflets. But when you're harvesting that fiddlehead this time of year, you're just harvesting the young leaves, the new growth of this year. And like see the whole length. Yeah. Okay. So let's look at the life cycle of a fern as a representative for the tracheophytes. Your book actually has a life cycle for all of the subgroups. I'm just kind of going with an overview for the major groups. So this is the gametophyte of a fern. Again, you would have a hard time seeing this in most ferns. It's probably about the size of your thumbnail, the gametophyte. It's not tiny, tiny. It's not microscopic, but it's pretty small. And it would be down in the soil, arising from the spore from a previous generation. And so this small gametophyte actually has both the female gametangium and the egg-producing gametangium and the sperm-producing gametangium all on the same little gametophyte. And so there's actually a section here on the gametophyte that is the sperm-producing section, and then here is the egg-producing section. And so in that same vegetative structure, it's producing both egg and sperm. So it doesn't need very much moisture for this transfer to happen within the same individual. We just need a few dewdrops. If we have multiple gametophytes and we have plenty of moisture, then you could also have exchange between gametophytes. So the sperm are going to swim over to the female gametangium where the egg remains stationary and fertilization occurs and the zygote forms. And then just like in our mosses, the zygote is going to actually give rise to a new structure. Only now that new structure is the sporophyte, a diploid sporophyte, but now it's going to look quite different and become the dominant visual representation, the dominant growth form of that plant. So a really young new sporophyte is going to actually have a shape that is different than the fern leaflet, and it will develop and grow into our mature sporophyte. And what most of you are familiar with when you're out looking at ferns or collecting fiddleheads is this mature sporophyte. The gametophyte is now gone. It's no longer present as the plant matures and lives multiple seasons. And it has this rhizome-like structure here that can actually send out new crowns and create new individual ferns from that structure. And so, yeah, yeah, they're dark. They look like a thick root underground. I was actually just digging up a bunch of ferns in my yard and I found a crown here and I dug probably about this far in order to pull up the next crown from that same individual. So they can actually send those structures, a couple of feet away from their body at least, and bring up a new individual. So they can have reproduction that way just by vegetatively reproducing from their structure. And then they can also do reproduction by releasing spores and having those new spores blow away, get carried to a new location, and start a new plant entirely. So when we were at the botanical gardens, you guys saw spores on a bunch of ferns. You've probably seen it maybe on your house plants or in a plant in the forest. The little capsules that produce the spores, the sporangia, are these dark structures on the underside of leaves. They are sometimes yellow or orange or reddish or brown. On some of the plants we looked at the botanical gardens, instead of being individual circles, they were more like patches of sporangia. All of those structures are producing spores. Those spores are haploid, and every single one of the spores that's released from that capsule can grow up and become a new gametophyte and start the cycle over. So again, you have a classic alternation of generations, half of the life cycle spent in a haploid state, half of the life cycle spent in a diploid state. We don't see this state much. That doesn't mean that it's not happening, but we do see this dominant sporophyte visually when we're out in the world examining ferns. Okay, does anybody have any questions about seedless plants? Seedless vascular plants? All right, I will go ahead and start our seed plants, which will maybe be more interesting. It seems like you guys are not so interested today. Not so interested. Pretty tired. Excited about salamanders. Aiming for salamanders. Is everybody really tired today? I feel like there's something about this week. Like I think it was Wednesday. Yeah, it was yesterday. My husband's like, it's Friday tomorrow, right? And I'm like, no, sorry. Nice try. Nice try. No, it's not Friday tomorrow. It's a long week. Yeah, it is now. It wasn't yesterday. It is today. I did not post these on Moodle yet, just because my day has been super busy and I forgot yesterday. So I will try to get them up this evening so that you have them. Yeah, they're all ready to go. I just need to post them. Mm, yeah, that was, I thought that they were both up. Yeah, no, but I just need to go. I need to actually write this on my hand. Quiz 7, quiz 7, and then 25 and 26 need to be opened. Okay, we'll try to do this. Okay, so we'll move on to chapter 26, which are seed plants. I will do one homework and one quiz for the two plant chapters together. They're about the same as the vertebrate chapter in terms of the total amount of content we're covering. So I will put them in together in one homework and quiz, and I'll try to get that up by tomorrow. So you have it for the weekend. So as we move into the next group, into seed plants, all of our seed plants, as we sort of foreshadowed in that previous table, are going to have a dominant sporophyte, and the gametophyte is going to be housed within the sporophyte. You're never going to see it. And they're going to have a much more terrestrial adapted reproductive system. They become completely independent of water. And so when we move to seed plants, it's like the difference between fish and amphibians and reptiles and mammals and birds, right? Make this transition to a reproductive cycle that no longer requires water. And so the seed is going to be that key feature, sort of like the amniotic egg was the key feature in our vertebrates that made a transition away from a moist environment and allows you to have a really successful lifestyle in a very dry environment. So whether we're looking at wildflowers that are starting to pop up right now, or a tree, just like with a fern, we're going to be seeing the sporophyte. And the gametophyte is going to be completely hidden in microscopic tissue that we'll never observe. Well, I wouldn't say never. We might take botany, right? You might observe it, but you would have to get a microscope out to see that tissue. So all of our seed plants are heterosporous. And if you remember from the earlier lecture this week, heterosporous, hetero means different. That means that these plants make different types of spores. So they make both microspores and megaspores. And I'm showing you pine cones here as the example. The microspores for pine cones occur within the pollen cone, the small cone, or the pollen producing cone of the tree. And those microspores are going to give rise to gametophytes. And so the actual gametophyte, instead of being a separate structure, is this, small microscopic structure embedded within the body of the sporophyte. In this case, embedded within the cone. And that gametophyte will then mature and develop into and produce ultimately the pollen in the microspore. So the microspore is going to give rise to pollen grains. Okay. Pollen grains individually by themselves are also very small and hard to see, but you can detect them in the environment. We're coming up on pollen season. Things are starting to pollinate out there. If you go out to your car one day and it's completely covered in yellow dust, right? That's pine sperm all over your car, just coated in pollen. That's the result of the microspores. And those are wind-borne structures or animal-borne structures that go out to find the product of the megaspores, which are the ovules. So this is a ovule producing cone, the ovulate cone of a pine tree. And the megaspores are in the cone. And those megaspores give rise to a single cell that is the gametophyte. And that gametophyte becomes the ovule. And then the ovule can be fertilized by the pollen to make the new individual. Okay, so there's a difference here in that the alternation of generation is still occurring, but this gametophyte generation is microscopic. It's actually housed within the body of the sporophyte. Okay, so the summary of that is that seed plants produce pollen and seeds. The pollen is the male gametophyte, which travels to the female gametophyte. Here on this dandelion, what you're seeing blowing away is not pollen, that is the seed. Okay, this coconut is a seed, right? The ovule is just the unfertilized seed. So the seed occurs when the pollen and the ovule come together to make a zygote. That zygote will develop within the original structure of the gametophyte and become the seed. Okay, so here is a seed with two different types of seeds, a pine seed and a bean seed. In both cases, the seed is a protective coating full of nutrients that houses the embryo. And so the sporophyte is the embryonic sporophyte is the small structure that will ultimately grow into the grass blade or the wildflower or the tree. And it is going to be housed within this nutrient pack. And all of this yellow structure here is nutrition, fats and sugars and proteins that the plant needs to develop and get started. And then it's surrounded by a protective barrier, which is going to prevent the seed from desiccating and drying out, and is going to protect it from damage until it begins to grow. Okay, we're going to skip the evolution of seed plants and just jump in to gymnosperms. The word gymnosperm means naked seeded. And so what this means is that the seed is not protected within the mother plant as it develops. It is on the surface of the structure that produces it. So the seed is actually exposed to the open air and the surrounding moisture and the external environment. Okay. The gymnosperms all have separate sex gametes that have to come together. Sometimes those are housed on the same plant. Sometimes they are separate individuals. So often if you're at a botanical garden, there'll be sort of the row of female ginkgos and the row of male ginkgos. You have to have both individuals producing pollen and egg in order to have fertilization. Probably, did all of you see the netophyte when we were at the botanical gardens? I think I pointed it out in the desert room. They also have several of them that are growing up near the bathrooms. I'm not sure why they're over there by the bathrooms, but they're reliably there. You guys saw the tall tree cycad, right? And then you're probably all pretty familiar with pine and spruce and hemlock and our other evergreen trees that are conifers. These guys are all in the gymnosperm group, and they're together because of how their seed develops in not in a protective container and typically on the surface of a cone. Mm-hmm. Mm-hmm. Right. There's just, I think that the ginkgo, like, I don't know this for sure. I think the Smith College ginkgo is a male because it doesn't stink. Stinky fruit. Yeah. Female ginkgo trees smell really bad. So like the alley behind a bar on a Sunday morning, kind of like pretty gross. So many botanical gardens have them, but they're not common in public places because people complain about how bad they smell when they're reproducing. I know, he's all alone. I know. Sometimes sexy smells. Um, so in gymnosperms, again, the thing that you see, the ginkgo tree, the welwitschia, the spruce, the hemlock, the thing you're seeing is the sporophyte, and the gametophyte is microscopic and housed within the cones. And before we do our like during our break, I'll bring over the cones. I meant to bring over cones. I have male and female cones of a couple of different species to show you guys so you can look at them. So this is a developing pollen cone, and this is a developing ovulate cone. And so the gametophyte is housed in these little circle areas where the seed is going to ultimately develop, and over here in these patches where the pollen is developing. Okay, so it's within the cone is where the gametophyte structure happens. Okay, um, here we can see the ovulate cones and the pollen cones right next to each other. So the ovulate cones have scales that contain a pair of ovules. And so if you look at a cone and you pull back the scale, at the base of it, there's two little grooves for each one of these individual brown scales. And those two little grooves are where the two ovules originally developed. And those two ovules will become two separate seeds. Okay, so the ovules are developing a pair for each scale or bract on the cone, and inside that section, in that little divot, is where the female spore mother cell is housed, and that gives rise to the sporangium, which gives rise to the gametophyte and the ovule, which is right here. The male cones or the pollen cones are smaller, and the pollen cones produce many, 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 many pollens relative to the number of ovules that the ovulate cones produce. And so housed within each one of these cones are these microsporangium pockets that are making hundreds to thousands of pollen grains per packet instead of just two per bract for the ovule. And then those pollen grains, when they're mature, are going to be released from the cone and they go forth on the wind. And so hopefully as they travel on the breeze, they land on the surface of these scales where the ovules are developing. And when that happens, the pollen lands on the surface of the ovule, and pollens actually send out a tube. And so when the pollen goes to fertilize an ovule, it actually grows a tube of tissue down to the ovule itself. And then the actual pollen grain transfers down the tube. It like tumbles down the tube and lands on the ovule and digs in. And so the pollen actually has to build a tube to get to the ovule. And that's one of the ways that plants actually can carry out sexual selection. So plants can reject pollen that are not the right size or they are of the similar genetics to themselves. And they actually can cut off the pollen tube or prevent the pollen tube from forming in order to reduce fertilization. So here we have our ovule, here we have our pollen. We produce the pollen by meiosis, we produce the egg by meiosis, and then we fertilize to get the zygote. The zygote is going to develop in the center of that pocket of sporangium, and that sporangium is ultimately the nutrient packet that's going to create the inside of the seed. As the zygote develops, it's going to start to divide and create this little baby embryo. And then when it gets to its necessary state inside the seed, the embryo arrests its development. It stops growing. And so the embryo is going to reach sort of its critical juvenile size, stop growing, and then the seed is finalized by formalizing this and reinforcing the seed coat and drying it out a little bit so that the embryo is protected in that little food packet, and that can then fall away from the parent plant. When that seed encounters optimal conditions for germination, like good moisture, warm temperatures, a certain depth within the soil, many seeds have to have no sunlight exposure to germinate. Some have to get high heat, like a fire. Others have to have a little bit of sunlight exposure, but the chemical signals in the surrounding environment are right. The seed will continue its growth. And so just like a young individual animal grows off of the yolk of its egg, the young embryo is going to grow off of that food packet in the seed until it can get itself above ground and start to photosynthesize. And then it's going to start producing its own food to continue its growth and become the mature sporophyte. Close, swapped out. Go ahead and do that during your break.